Okay, welcome everyone. So it's a real pleasure to have with us Dr. Levine here in the distinguished Josep Carreras Leukemia Research Lectures. Dr. Levine, he is the chief of the Molecular Cancer Medicine Service, Human Oncology and Pathogenesis Program at Memorial Sloan Kettering in New York. He's also the Lawrence Joseph Dainin Chair in Leukemia Research and Professor of Medicine at Weill Cornell Medical College. Dr. Levine has an extensive background in medicine with training at Harvard, Johns Hopkins, Mass General Hospital, and Dana-Farber. And his studies has been always in the genetic basis of malignancies. He has done a lot of work understanding the JAK-STAT pathway, and in the recent years in the mutations in epigenetic modifier genes, particularly in the context of clonal hematopoiesis, that, as you know, is one of the hot areas now in hematology. He has received many recognitions from his work. Just to mention a couple, the Dameshek Prize from the American Society of Hematology, he's a Scholar Award from the Leukemia and Lymphoma Society, and he serves in different scientific advisory boards. So it's a real pleasure for all of us here in Institute and abroad to have him with us. And the title of his talk will be Genetics and Therapy of Clonal Hematopoiesis and Myeloid Malignancies. Ross?
Thanks, Manel. It's really a pleasure to be here today virtually. And I'll just put my disclosures up briefly, none of which are relevant to my talk today. And I'll just say personally that it's a pleasure to give this talk, although I wish I was able to visit in person. You know, Manel and I have a long history, and in fact, my second paper of my career was a paper that Manel wrote that I was able to collaborate on, and it's really a pleasure to come back some 23 years later and give a talk. And we're excited, actually, hopefully, to build some collaborations together between our laboratory and the science here at Sloan and your institute. And I think there's a lot of opportunity for that moving forward.
Thank you.
The other thing I'd say before I talk about science is that we're in a interesting and challenging time. I think for those of you that are listening to my talk that are early career investigators or trainees, we know it's really hard. We know it's challenging to know what's going to happen to your careers, to the job market, to funding, to your ability to do science. We know many of you are getting back to work after the pause for COVID. And I just want to make the point that we all want to support all of you with everything we do. To those of you that are on the clinical front, thank you for all you've done and for all you do. And for everybody, please stay safe. And the last thing I'll just say, I think, on that is that it's important to remember that it's really hard to be productive with online science, with kids at home, with in and out of lab. None of us planned for this, and we realize that we're working under less than optimal context. But it's important to stay connected, and I think things like this are really important to continue to have lectures and really maintain a network. And that's actually more important than that paper or that grant. And I can tell you that the important thing is just to get something done and to find new ways to contribute. And as some of you may know, if you follow me on Twitter, you'll know that I feel strongly about this. And we're all in this together, and we're in this really to make a difference. And I'll start with an acknowledgement slide and make the point that all the work I'll talk today was done by some amazing trainees in my lab, Bobby, Lindy, Michael, Aaron in particular, with help from Tiff and Nicole and Richard. We've got great collaborators, including Jason and Sarah and Martin and Giannis and Todd Druley. So my lab has been focused on acute myeloid leukemia predominantly for the past decade, and I'll just make the point that, you know, although we've improved outcomes, we've not done so dramatically. Those of you will know that one of the late-breaking abstracts at EHA this weekend was the venetoclax azacitidine, which showed an improvement in overall survival for older adults with AML. I'll remind you that the overall survival for the venetoclax aza arm of that trial was 14 months. And the majority of people who got ven-aza died of their disease. So we've got a long way to go, and we need new insights biologically, we need new therapies, and we need to understand how AML is different in every single patient, and also within individual patients, and I'll talk about that in significant detail. But although AML is a complicated disease to understand, it's got a tractable genetic fingerprint. We know there are only 50 to 70 recurrently mutated genes for AML genome, and there are only 3 to 8 coding mutations for AML genome. And so that means that a small number of mutations and a small number of genes give us great clinical and biological complexity, and that's where my lab has really been focused and will continue to focus. And in the past few years, as Manel said, I think we've learned a really important observation from many labs, including ours, about the concept of a pre-leukemic state, which we now call clonal hematopoiesis. We know that mutations in largely epigenetic regulators, P53, JAK2, can occur in the absence of overt malignancy, and these can be a precursor state to transformation. The implications of this on the genomic profiling are expanding, how we follow patients, what it means, how we track people's response to therapy. We know from data from Ben Ebert and others that it increases the risk for cardiovascular disease. But I think what I'm most excited about is the idea that this may represent the first frontier for effective malignancy prevention. And the idea that if we can identify these mutations, figure out what they do, and intervene, we might be able to prevent overt transformation. And this slide really lays that out. The idea that there are a lot more people in the United States and worldwide with clonal hematopoiesis, millions, than have overt myeloid malignancies. And that means that if we could intercept this process in the middle, we might be able to prevent the overt development of disease. And I think in the long term, that's our highest likelihood of achieving success. And so from our lab standpoint, we want to understand 3 things. We want to understand what are the early events that initiate this process? What are the events that lead to transformation? What mutations, what order, what cell type? And then once you have overt disease, what are the dependencies in that process? And how much of that is governed by the journey a cell takes to become a leukemic cell? And the way I like to think of it, and I don't know if this is popular in Barcelona, I would hope it is to think of leukemia and cancer in general as a Jenga puzzle, that we want to understand what are the events that you have to build to give you AML, and then what are the ones we can take apart so that the AML comes crashing down. And so that's really been our major focus and will be for the next 5 years at least. And so we've learned a lot from genomic studies of AML, particularly using bulk sequencing, that lets us know what are the mutations and generally what are the order of mutations. We can use variant allele frequency as a way to predict what are early events like epigenetic regulators and JAK2, and what are later events like NPM1 and then RAS, FLT3, and occasionally P53. The challenges, though, are that this is bulk sequencing. It doesn't tell you how every clone is evolving over time. And so that's led us to really focus on this concept of deciphering and elucidating clonal evolution during myeloid transformation. Can we better delineate the sequential acquisition of mutations? Can we understand what are the key genetic events that promote transformation and clonal expansion? Are there different trajectories to AML based on the different mutational subtypes? How does AML evolve not as one clonal trajectory, but a milieu of clones? And can we link all of this to therapeutic dependencies? And so we think that by doing single cell mutational profiling and not just RNA profiling, we can understand that with greater granularity. And so Lindy and Bobby in my lab have set out to do that by developing single cell mutational profiling of AML. All of this work is available on bioRxiv, and there's similar work from Koichi Takahashi, and I'm happy to talk offline or on Twitter or by email about the results we have. We've done about 146 samples. We have a custom platform that allows us to query a subset of genes, and we use the Mission Biotapestry platform. And then I'll talk a little bit later on about using DNA plus surface immunophenotype to look at lineage trajectories. And so what we get for every patient is a clonotype. And the clonotype gives us, for every patient, different combinations of mutations and how many cells have each permutation of mutation. So in this patient, you could see the dominant clone is an IDH2 STAG2 NRAS mutant clone, and that's a little bit different than some of the minor subclones. And so once we did that, we were able to look at the number of mutations, at the number of clones, and clonal diversity in AML. So as you go from clonal hematopoiesis to MPN, to AML with and without mutations and signaling effectors, the number of mutations go up, but the number of clones goes up even more than the number of mutations. And what that means is that it's not just acquisition of mutations in one clone, but clonal diversity. And we think that's an important lesson. And so what do we learn if we begin to look at different disease subtypes? So clonal hematopoiesis in general, in most patients, about 80%, there's only one mutation, so it's not clonally complex. But in 20% of patients, we see more than one mutation, and those patients are at higher risk for transformation. And so we looked at a group of patients with clonal hematopoiesis that had more than one mutation, and in most cases, we see that if you see in this patient that had 3 different mutations, it was in 3 distinct clones, although one clone did acquire a second mutation. So we believe that clonal hematopoiesis is the evolution of multiple different clones over time. By contrast, AML is a disease of both clonal branching and clonal evolution within clones. And you can see this patient's a great example, where you have a dominant clone that has DNMT, IDH, and NPM1, and then, lots of distinct subclones. And in fact, in 86% of our patients, we either see 1 or 2 clones that really are the bulk or dominant clone. And what that means is that although there's lots of antecedent clones and branching, there is what we would call a winner clone or a dominant clone. And this clone either has an enhanced proliferation or self-renewal advantage, or may in fact secrete factors that discount or reduce the fitness of other clones, or both. And we think this is an area that many labs, including our own, hopefully will study in the next few years. We then wanted to get an enumeration of clonal diversity, and we used the Shannon Diversity Index, and you can see that the clonal diversity increases more than the number of mutations. And in fact, as you go to more advanced leukemia, although there's a dominant clone, its size is smaller. And what that means is, as leukemia evolves to more advanced or adverse risk, there's a dominant clone, but it's less dominant, and you get many different branching subclones. And that means that we think that there's different patterns of clonal evolution and dominance. And that led us to wonder, are there different roles for different mutations in establishing and maintaining clonal dominance versus clonal branching? And in fact, different mutations have a different likelihood for being in the dominant clone. And you can see some mutations like IDH and NPM1, when they're present, they're virtually always in the dominant clone. Whereas other mutations like NRAS or FLT3 are actually very infrequently in the dominant clone. And this means that you have very interesting factors where you have some mutations that are almost always in the dominant clone, some mutations that are almost always in subclones. Then you have TET2, and TET2 actually can play both roles. Sometimes it's in the dominant clone, and sometimes in minor clones. The problem, though, with these analyses are they look at individual mutations by themselves. What we really care about are cooperating and exclusive mutations. So we began to look at inclusive and exclusive mutations, what co-occurs and not. On the left, this is using bulk sequencing. You see some co-occurrence and some not. But on the right, when you look in cells, you see much greater matrices that show greater co-occurrence and exclusivity, which I think is really important. So we began to look at this with some granularity, but with the caveat that 150 samples and 500,000 cells is not enough for us to be able to do this. We're going to need a lot more data. And so we've seen a few things that have taught us some things. The first is that when we see epigenetic regulated mutations and they co-occur in a patient, they're almost always in the same clone. So you can't think of what does DNMT3A or what does TAT2 do. You have to ask, what do these two mutations do together? And you have to look at all of these different permutations and how they collaboratively co-opt the epigenetic state of stem and progenitor cells. By contrast, epigenetic signaling effectors are almost never in the same clone, with the exception of JAK2 and NRAS mutations. And what that means is that when you think of epigenetic regulators, they're in the same clone. When you think of signaling effectors, they're in distinct clones. And in fact, when we begin to put it all together, we see different patterns of co-occurrence. So DNMT and IDH can co-occur in the same clones, but when they do, they have different signaling effectors. So DNMT IDH mutant clones, if they're IDH1, look like IDH1 clones with different mutational patterns. But DNMT3 IDH2 mutant clones have a different co-mutational pattern than 3A only clones. And JAK2 and NRAS tend to occur only in IDH2 only clones, and FLT3 mutations tend to occur in 3A IDH2 double mutant clones. So we think you could begin to build, triads and maybe even 4, 5, 6 mutations that occur in distinct clones. And then we wanted to begin to look at the trajectory, not only looking at who co-occurs, but who promotes clonal expansion. And so this takes advantage of the number of cells with different mutations. So look at this patient on the left. They started with a DNMT3A mutation, but the clone was still small. They acquired an IDH mutation, the clone was still small. But once they acquired NPM1, the clone dramatically expanded. And you can see the same thing here, IDH, IDH FLT3, IDH FLT3, NPM1, and that clone is much bigger than the one that is NPM1 and NRAS. And so this lets us ask what mutations are initiating versus driving clonal expansion. So if we ask what mutations are initiating, you see 4 different patterns. You see that IDH, DNMT3A, JAK2, these are usually the first mutation. FLT3, RAS mutations are always late events. NPM1 and ASXL1 are actually early events, but never the first event. And so NPM1 is usually the event that happens on top of the epigenetic regulator to cause clonal dominance. And then TET2, like before, can be early or late. And so can we prove, that was just based on prediction, based on the genomic data, can we see these initiating clones by looking for single mutant only clones? So the DNMT3A mutations we see are in the early clones, and we see evidence of single mutant clones, whereas the PTPN11 and RAS and FLT3 are late events. And for DNMT3A, we see interesting things, like R882 only clones are actually harder to detect than other DNMT3A mutations. Is that because there's difference in leukemia initiation versus maintenance for different 3A mutations, or because the R882 clones, when they acquire that mutation, they quickly transform to disease? The answer is we're going to need 10, 100-fold deeper sequencing at single cell to really look at this, to get greater look at architecture. But the lesson for us is that different combinations of mutations cause clonal expansion, like DNMT3A, IDH, or NPM1, FLT3, and other combination mutations. They're in the same cell, but they don't drive clonal expansion like NPM1 RAS. And so if you want to study the biology of AML, you want to study the mutations that together promote clonal expansion, and that's an important point. We then wanted to look at different samples over time. So this is a patient who we had 2 samples in the MPN phase and the AML phase, and you could see that the MPN phase in red was largely CALR ASXL1 mutant, but when it went on to disease, it acquired the IDH1 mutation, but that clone was actually rare and present. So it's not like acquiring the mutation, boom, you get AML, but that's the clone that when additional potentially epigenetic remodeling happen, gives rise to overt AML. So we think that understanding the genetic and epigenetic determinants of this clonal switch and transformation is critical. We've looked at people that had AML that, for example, had FLT3 mutant AML and got targeted therapy. In this patient, you went from the red to the blue. You could see that the RAS clone was pre-existing and this one grows out, but you get a bunch of other RAS and RUNX1 clones that grow out at acquired resistance. But this patient, I think, tells a very interesting story. The same kind of story. They had a FLT3 mutant disease with lots of different subclones. And then when you treat, you get 2 different groups that grow out. One was an antecedent clone that actually had a RAS mutation that now grows out, and another were all of these minor RAS clones that weren't there at all or were very rare and now grow out. So you see 2 branching clones that grow resistance. It's not like you go from this clone to that clone. It's this population of clones to this population of clones. Some of them grow from one to the other, and others are de novo, sweeping and acquired. The challenge, though, is how do we go from this interesting genotypic data to phenotypic data? So the next thing we've done with our colleagues at Mission Bio is to begin to marry mutational data to cell surface data. And so we do this by having oligoconjugated antibodies that allow us to look at this in different cells. And so this is what we get. This is a patient with AML, and we can show all of these different surface markers versus different clones. All of the mutant clones are here, and then these cells are wild type, and they're all CD3 positive, so you can detect the non-mutant CD3 cells. And so what can you do with mutations and immunophenotype at the same time? So in clonal hematopoiesis, you can track a mutation versus lineage. So this DNMT3A mutation was in the myeloid cells and in the T cells, but very rarely in the B cells. And that is a good example of looking at lineage output of mutations. Whereas here in this patient, you have two different DNMT3A mutations, and one of them, the canonical R82, is in the myeloid and B cells, and one, this R635, is in the T cells, but not really in the B cells and only rarely in the myeloid cell. So we think for clonal hematopoiesis, it's going to be very instructive to take different mutations, different combinations of mutations, different clinical scenarios, and look at the clonal or lineage output of different clonotypes. In AML, we could do it much more complex and interesting things. So this is a patient that had lots of different clones, including FLT3 and RAS mutant clones. And what we saw were that the RAS mutant clones had expansion of this orange subset. And this orange subset actually was CD11B positive cells. And in fact, the RAS mutant cells in this patient were virtually all of the CD11B cells. So imagine if you had a CAR T cell that could target the RAS clones, which can cause acquired resistance to targeted both IDH and FLT3 inhibitors. So you could combine a lineage-specific surface strategy with other therapies. And we think this is the future of AML, to marry genotype to immunophenotype and to develop cell surface strategies that might not treat the whole clone, but might treat recalcitrant subclones. And we can actually show that in 8 of 14 samples, the dominant clone had a immunophenotype that was different than the minor subclones. So we think marrying the genetics and epigenetics to the surface expressed proteome will be a great way to understand both biology and maybe find new therapeutic targets. And we can begin to look at how different combinations of mutations promote different lineage marker expression. So NRAS and IDH1 lead to CD11B expression, whereas DNMT and IDH1 lead to CD34 expression. And so we want to move forward and do lots more in this direction, and we're happy to talk about this, including Pablo Sanchez Vela, who's from Spain, who's working with Manel and I, who's going to look at this in solid tumors with clonal hematopoiesis, because you can actually use the cell surface to fractionate the tumor from the CH, and we think that'll be a fun way to do this without separating all of these populations. And so in summary, for this first part, we think that single-cell DNA sequencing of this single large AML cohort lets us look at clonal architecture and disease evolution at single-cell resolution. We can see evolution and increase in clonal diversity over time. We can see how different mutations lead to disease initiation, clonal expansion, and subclonal propagation. And we think that you can link surface mutational repertoire to cell surface and therapeutic response. And obviously, there's a lot more to do here. We're happy to talk to folks about this technology moving forward. So the next step for us is to marry this human data to biology in the lab. And so the other thing we're doing in the lab is now going back and saying, if we see different orders of mutations in the lab, can we model this in the lab? And this is an example where in our genomic data, we know that the NPM1 mutation occurs before TET2. So we then make mouse models where we do NPM1 and TET2 in the different orders. And if we engineer it in the right order, Lindy's done here, you actually get an overt, fully penetrant AML that you don't get with the wrong order, where you do TET2 first. And in the long term, what we'd like to do is to build a series of genetic tools that let us order different mutations and induce activation in different cell types, and actually go back to the lab and study the biology using functional models of what all of the single cell data teaches us. And what Bobby in my lab has done is to begin to develop those tools, and I'll just give you one example, which is a FLT-inducible FLT3 model, where now we can induce FLT3 after all of the other genetic events, exactly the way it occurs in patients, unlike current FLT3 models, which are germline, which occur first. And this model works very well. And we've induced it by itself. We get myeloproliferation that's much more penetrant and significant than if you have germline, suggesting there's feedback if you activate FLT3-ITD developmentally. But most importantly, if we do it with NPM1C in the right order, we get a fully lethal penetrant AML that's much more rapid than George Vassiliou's elegant model of NPM1 and FLT3, where FLT3 occurs first. And this model has a CD34 negative immunophenotype that actually mirrors the human disease. And we're doing lots of work with this model to understand its biology. And then we can do lots of other cool models with NPM1, TET2, DNMT3A. So we'll have a lot more to say about modeling clonal evolution in AML. And maybe if I visit Barcelona in a year or so, I can give you guys an update on that. But the fundamental thing is that, we need to understand what these mutations do alone and in combination at greater granularity. And so there's a lot we don't understand about how this all starts. how do we understand the process by which these early mutations initiate and maintain disease? I would argue that we don't understand how these early mutations change the fitness of hematopoietic stem progenitor cells. We don't understand why only a very small subset of people go on to overt disease and others don't. We don't understand how to target clonal hematopoiesis. And we think there's a lot to learn about aging and cancer. And we think this is an area, I'm sure, that's of shared interest between our institute and yours. And so if we take a step back and ask, what have we learned from epigenetic regulators? We know that these mutations in DNMT3A, TET2, ASX1 lead to increased stem progenitor cell fitness and myeloid bias. They have variable phenotype in vivo and self-renewal. They are relatively faithful, but they're mostly in mouse models. We need better human systems. But we don't know what the downstream targets that these epigenetic regulators induce to lead to increased fitness, although some really cool work, including recently by Tan Landau, has given us some insights that might lead us down that road. One major limitation in the field is that we don't have ways to culture these cells that allow us to keep them in culture for more than 2 to 3 days. And we need assays that read out self-renewal and differentiation, not just survival. And we need to use them as a platform for therapeutic target discovery. So my lab about 3 or 4 years ago said we need systems to culture leukemia cells and understand their biology at longer duration and more faithful phenotypes. And this is not different than the organoid system that Howard Hans Klebers developed. And so we tried a whole bunch of systems. And the one that we've sort of settled on is a system developed by Jason Butler and Jaheen Rafi, where they culture hematopoietic stem and progenitor cells on an AKT expressing stromal layer, which they showed on the left can allow stem and progenitor cells to maintain in culture, but more importantly, allows them to propagate in vivo after extended ex vivo culture. And Jason's done some really elegant work in MLL models showing that they work. So my lab decided to replicate that work, and we were able to show that we could maintain stem cells ex vivo and give rise with about 500-fold expansion of stem cells. They can transplant secondary and tertiary. They get about, again, an expansion of stem cells, but also lots of differentiated cells. So this culture, we think, recapitulates a lot of the stem and myeloid cell microenvironment and gives us a way to study the biology of these cells long term. But we're not a wild type stem and progenitor lab. What we're most excited about is the idea that you could study mutant and wild type cells in different contexts. So in this experiment, what we did was culture TET and 2 mutant and wild type cells in the same dish. And we could show that we can map where TET2 cells expand and where wild type cells expand. So you see much more TET2 mutant cells in the more primitive cells. Although not in the most primitive stem cells, it's actually slightly more distal. If you inject that mix into mice, the TET2 cells not only win out, but they have myeloid bias, exactly as you see. So we now get a 2 to 3 week readout of something that took us 6 months to a year to read out before. We've done the same for ASXL1. Here we see the expansion in more primitive long-term stem cells, much more so than differentiated cells. And we've done single cell RNA sequencing to show that in the mutant cells, in the stem cells, we can show there are differences in expression of genes, including myeloperoxidase, Ly6A, and Ki67. And we have a lot more to say about what are the downstream gene regulatory pathways induced by these mutations in different cell types. But I'm most excited about the idea of using them as a screening methodology. And so this just really makes the point that what we can do is take TET2 or DNMT3 or ASXL1 versus wild type cells and culture them in different drugs. And you can recognize a lot of leukemia drugs on a list. Any drug that kills the stroma doesn't get used. And so we've begun to screen different hematopoietic cell types and genotypes for chemical dependencies. And the other thing we're excited about is that you can do genetic manipulation in this system. And so what we've done in this system is show that we can knock in and knock out different genes ex vivo and maintain their biology. So we can take a TET2 knockout and knock out TET2 ex vivo and show that you get expansion of mutant cells, and then we can actually put them back in the mouse. And so again, the idea that you can do genetic manipulation in this system. So this is all good and well, but chemical screens, we can only do 10, 20, 30 molecules. This knockout strategy is only good for genes that we know about. We want to scale this up and out. And so I thought I'd end by showing the newest experiments from my lab that we began just before COVID. And that was the idea that could we expand to doing agnostic screening of all of the different leukemia and clonal hematopoiesis genotypes. And so Mike Wartz, who's a grad student in my lab, set out to say, can we do CRISPR screening or CRISPR editing in this ex vivo system and then retain those cells ultimately in vivo? And so what he did on the top was take Cas9 mice and try to edit out CD45 using a guide. And you can see that he gets about 70% editing when he directs to a specific guide. That's pretty good. And he can maintain this for weeks at a time. So he's successfully edited out this gene, and you can actually track these cells over time. The problem with this is then you need to cross every single one of your mice to a Cas9 mouse. That's not ideal. So then he had the idea, what if he takes wild type cells and he electroporates in either Cas9 protein or mRNA with the guides, and you can see he still gets 50% efficiency. So this essentially opens up the whole toolbox. Now you can take a TET2 mutant mouse that may or may not already have TET2 deleted or excised. It depends on whether you want to do CRE and Cas9 ex vivo. You can expose it to Cas9 and your set of libraries and now do CRISPR-Cas9 screening. And so that really opened us up right before COVID to doing that. And so Mike did his first tests in January and February. So this is all new data. And what he did was take an epigenomic library of a set of targets developed by Chris Valkich, and he looked ex vivo culture, and he could maintain between day 7 and day 1, you could see a correlation of 0.996. So we maintain representation of the library. That's great. And he gets a reasonable number of guides in per cell, so he's not over-infecting. And so this really emboldened us to begin screens. And so the first thing we did was take wild type cells and do CRISPR-Cas9 screening of an epigenetic library. On the left, this is without Cas9, you get no hits, so the screen is clean as a whistle. On the right, this is by adding the Cas9 mRNA as delivery, so not crossing the Cas9. And you can see that most of our guides don't score, they're innocuous. You get a few that have positive enrichment, and those are very interesting. But I think the most interesting thing was that you get a series of depletions. And we see this both at day 7 and day 14. We're still trying to figure out the right time kinetic, and we're injecting all of these in vivo into mice to see if the in vivo readout is different. So this to us is very intriguing. And so what do we see? What are those targets? And so what was fascinating was that we layered our targets on against Junwei Shi and Chris Valkich's targets in MLL cellular models. And you could see that a number of the things that are purported to be MLL dependencies like BRD9 and P300 are shared by wild type stem cells. I would predict that all of these are pan-hematopoietic dependencies. By contrast, you see that we found 4 genes that are only required in wild type stem cells, but not in the MLL model. So those are interesting. But I think most importantly, we could identify and validate, including HBO1, a recent Nature paper by Mark Dawson, that these are leukemia-specific dependencies not shared by hematopoietic stem cells. And so this is what we're going to now do at scale. And so again, what we're going to be doing as we get back to work and we can't travel is taking all of our leukemia genotypes and screening them. And we're looking for things that score in leukemia, but not in wild type stem cells, or score in defined genotypes. And we're very excited about that. And then you can do lots of things, compare pre-leukemic to leukemic. You can add a drug and do sensitizers, lots to do here. And we've got plenty of work to do, and we'll share it in real time as we do it. And we obviously believe in this idea of open science, so we'll have a lot to share, we hope, in the next year or two. So in conclusion, we believe that myeloid transformation can initiate from these early events, and you get progressive evolution to overt myeloid malignancies. We can map the cascade of genetic epigenetic events that drive myeloid transformation. We're excited to work with Manel using methylation profiling in a lot of these different systems. Single cell profiling gives us a look at clone-specific genotypes and phenotypes, and lets us look in novel insights into the leukemic ecosystem of different mutant clones. We can model clonal evolution with increasing accuracy and develop better preclinical models. And we think novel preclinical systems can be used to identify genetic and pharmacologic dependencies, and we'll have a lot to say about them moving forward. So with that, I'll just make the point that Bobby and Lindy did all the single cell profiling work. Bobby developed the whole BMEC system, and Lindy and Bobby have done all the order and developing of new animal models. Mike has done all of the work with the CRISPR-Cas9 screening. Pablo is really doing all of the CH and solid tumor work. And Aaron really began the work in single cell emission bio with Bobby and Lindy. Tiff and Nicole have been incredible helpers, as have all the other technicians in our lab. Richard has been incredibly helpful with all of our informatic analyses, and Jason and Sarah and Martin have been incredible helpers in lots of this work, and Giannis. And we worked with Todd Drewley more recently on single cell error corrected sequencing, and the folks at Mission Bio have been extraordinary collaborators in all of this work. With that, I thank you, and we've got, I think, plenty of time for questions. So thanks again, Manel, for hosting me, and I'm happy to take questions. Thanks so much.
Thank you very much. Thank you very much. Ross has been wonderful, and thank you for the memories also. So it's huge amount of data, so it's difficult to. I think it is very relevant, and many aspects that there are many questions here. So let me read one here, for example. Let's see. Yes. What do you think about here in these evolution models that you have? It can be some neutral selection as opposed to Darwinian selection in some of these studies you have done in single cells.
It's a great question. I think there probably is a lot of neutral selection occurring in evolution. I think my view is a lot like Darwin, that, and like Stephen Jay Gould, who talked about punctuated equilibrium, that there's a lot of neutral evolution and drift happening, and then there are moments of selective pressure, and that then puts that on top, and that a lot of that variation's occurring with very little selection, and then that occurs. That can be everything from in the setting of infection or stress. That can be a therapy. That can be inflammation from obesity, from smoking. I think we're going to learn that there are these moments where there are what I would call bottlenecks that are challenged. And in those moments, specific clones win. But in general, that's occurring in a backdrop of a lot of neutral evolution and neutral evolution and drift.
Yeah. Another question, it says that we have seen in your slides sometimes CHEK2, mutations in CHEK2. What is known about the role of DNA damage repair genes in overall in AML?
Yeah, I think the answer is not nearly enough. You know, P53 has long been thought of as a solid tumor gene. I think honestly, and this includes my own work from 7 or 8 years ago, if you look at de novo AML in younger adults, a lot of the mutations of DNA damage genes, P53, CHEK2, and others, are quite rare. But if you look at AML, when we see them in the hospital, and older adults, secondary AML, therapy-induced AML, clonal hematopoiesis-involved AML, there's a lot of mutations in DNA damage pathways. And what the mutations are doing, what they are, and how they functionally contribute, I think we barely understand the early principles, and we need a lot more work. So I hope some of you will really dive into that. It's a great question.
Yes, thank you. I remind all of you that you have to send your questions using the application here online, okay, in written form. So I have here also 2 general questions that are appearing now. One, it says about the clonal hematopoiesis. It is known that sure the impact in cardiovascular. So what about beyond cardiovascular? Do you think other human disorders are going to be affected by clonal hematopoiesis, by CHIP?
Yeah, I think they will be. I think that the challenge is twofold. One is that you need large cohorts with annotated information about other diseases where there's been banked or genotype samples. And for cardiovascular disease, there's just been so much work that it was easy to leverage that. But my prediction is that most diseases of the aging population will have some component where clonal hematopoiesis makes them worse. And the rate limiting step, honestly, is we need young, smart people to think about all these diseases and study it. And I'm hoping that many of you will do that. And I'm excited that the field will do that. And I just want to enable other people to really do that moving forward.
Yeah, and talking about excitement, in AML, are you expecting any big breakthrough that really this survival is not so good is able to make a difference?
You know, I sure hope so. I think we've had after 40 years of very little benefit until 2016, we now have many drugs approved, the IDH1, IDH2, FLT3 inhibitors, venetoclax, the hedgehog inhibitor. But again, I have to say, following EHA for the last 3 days, I think we've had progress now, especially in higher response rates and prolongation, but we have a long way to go. And I think a 14.5-month survival with Ven/Aza and AML is not nearly good enough, and we need to do a lot better. And so I think we have a long way to go before we can declare significant success.
And what about clonal evolution in relapse cases? What do you know about this? What have you studied there?
Very little. We made the decision that because this technology is new and complicated and it's expensive, that it would be better. I think we thought it would be better to study a large number of patients at one period of time. The next step is to start doing diagnosis and relapse and looking at that. But we thought that would be a study of another 100 or 200 patients of its own, and we wanted to start somewhere, but it needs to be done.
Yeah, and if at the end you have this poor prognosis overall, so what do you think is the relevance of stratifying patients? It can relate to the drugs, but for prognosis, pure prognosis.
Well, it's a really interesting thing because I think if you take the data that I talk about and everybody else about clonality and heterogeneity, you would think that the stratification was doomed to fail because you'd say it's all complicated, every patient's got 20 clones. But the reality is that if you look at the dominant genotype and the big genetic and cytogenetic alterations, they have prognostic value for chemotherapy, for transplant and targeted therapy. And what that means is that there's a lot of biology in the dominant clone and that the minor clones are a factor, but they're not so complicated that they always drive all of the biology. And so we have to always be realistic that there is an important role, even for standard genetic profiling without fancy single cell in predicting outcome. And even FLT3. FLT3 is a late event in AML, but it's powerfully prognostic. And what that means is that there's biology there, some of which we understand, some of which we don't. That if FLT3 is there, it's bad. And why it's bad, whether it's in the dominant or something, we got to figure that out. And that's an important area moving forward.
Yeah, because I don't want to take a lot of your time on a Monday. So I'll take the last question here, that have you done any synthetic lethal experiments? Because you have to inactivating mutations in 2 different pathways.
Not yet. We think that's a great area. I think that's one of the areas we'd like to explore moving forward. You know, we've done mutual exclusivity like IDH and TET previous, and with Ari Melnick and Craig Thompson, that was one of the links for genetics and epigenetics, but I don't think that was a true synthetic lethality. We believe that some of the CRISPR technologies and the ability to model different mutations with different recombinases will let us test synthetic lethality, and we'll use the human data to guide those experiments. So lots to do there and lots to figure out, and we hope lots of us will work on that.
Yeah, so I'll take advantage of being the chairman just to ask the last question. So what are the IDH inhibitors? What is the current status of those?
Well, they're approved. They have about a 20% complete response rate as single agents and a 40% response rate. There was data presented at EHA by Courtney DiNardo that if you combine the IDH inhibitors with hypomethylating agents, you probably get a 50% response rate with a pretty reasonable tolerability profile. But that's interesting because what that means is that 50% of the IDH mutant patients aren't responding to pretty potent epigenetic therapy. I would posit, I don't know what you would say, Manel, that if you had an IDH inhibitor to a hypomethylating agent, you would think that the methylation remodeling would be maybe not fully altered, but pretty densely altered. And that means that there has to be, in different patients, different dependency on that epigenetic remodeling, and there's something we don't understand. And we think code mutations matter like RAS and FLT3, but there's probably biology there that'll be fun to figure out together, and that I think it's not so simple.
Okay, it has been wonderful, as I said. No more work on a Monday at this stage. So thank you again for your presence and your lecture, and please join me to clap, Doctor.
Thanks so much. And like I said, next time I'm in Europe, I look forward to visiting everybody in person. So I'll see you guys soon.
You're more happy to be here. Thank you.
All right.
It's been great. Thanks.
Thanks.